and relaxed during the break, so we can start with little crash course into biotechnology. I would like to welcome you to the little crash course into biotechnology, or how to make a glowing fluorescent fungus using the knowledge we gathered in our biotechnology studies. So, let us start with genetically modified organisms that can glow when they are put under specific light conditions. We call those fluorescent organisms. So, as we can see on those photos, they look quite nice. They have nice colors. They're quite spectacular, let's be honest. So, how can we make them in the laboratory? So, we can see that those glow. How? As always, the answer, as most things in life, is physics. And now I need you to bear with me, because this schematic is a little hard, but when you understand it, everything will be very easy for you to get. So, this diagram shows spectrum of electromagnetic radiation. The thing I want you to remember, because there will be a little test later, is that the shorter the wavelength of electromagnetic radiation, there's more energy in this wavelength. For example, gamma radiation. And the longer the wavelength, there's less energy. And visible light spectrum is also electromagnetic radiation. So, by knowing that, we now know that violets have more energy than, for example, reds. Yes? Great. So, now, how fluorescence works. Here we can see Domański diagram. Don't worry, you won't have to remember it. It's just a way for scientists to explain how this process works, but it's just 3 steps. So, we have this molecule here, and it has ability to absorb some energy. So, we heat it with blue light that has some energy. So, this molecule becomes energized. Then, it cannot hold this energy for long, so it loses a little of it. Then, it has to emit the energy it gathered. But, as I said, it lost some of it along the way. So, I have a question for you. If it was hit by blue light, here you have a little cheat sheet for the question, what kind of light will it emit? Anyone?
Green?
Yes, it will be a green light. So, this is how fluorescence works. And the substances that can exhibit fluorescence we call fluorochromes. And now, for the little harder part, after we understand the physics of it, how do we make things glow? So, as I said, there are genetically modified organisms that can be fluorescent. But in nature, there are also organisms that can be fluorescent just by themselves. They just like that. So, for example, this beautiful jellyfish or this coral called Discosoma that can be bought for aquariums. So, those organisms have proteins that are fluorescent. For example, jellyfish has green fluorescent protein called GFP, and this coral has red fluorescent protein. So, scientists thought, wait, so these organisms have proteins that can glow. What if we put those into different organisms? Will they also glow? Yes, but how do we do that? So, as all you may know, we have DNA. All living things have DNA. DNA is the material that gathers all of the information that's required to make a functioning organism, and all And each this information that contains one function, we biologists call genes. So it's like a recipe. For example, a recipe for fluorescent protein. So we have the genes for green fluorescent protein, for example. So what do we do with it? And there comes the another process biotechnologists really like to use, called genetic transformation. This is a process of taking gene from one organism and putting it in the different one so it can get the protein, function, trait of the other organism. This is not only laboratory technique, but it also happens in nature all of the time. For example, with bacteria. We have here the example of soil bacteria called Agrobacterium tumefaciens. This bacteria attacks plants. You may sometimes see on plants some like those kind of tumors. This is a home for bacteria. So it uses genetic transformation, so plant will make a nice home for them, it will produce them food. So when plant is injured, Agrobacterium enters it and gives this gene, this recipe for tumor to the plant. And plant does all the work for the bacteria. Bacteria doesn't have to spend any energy later. Plant just makes home for it, makes food for it. It's very nice for bacteria anyway. So this is how this process looks in schematic. So as I said, DNA is material that gathers all the information about functions and organism, but it can be present in different forms. You may know about chromosomes. We have chromosomes as humans, but DNA can also be in different forms. For example, those little circular forms that we call plasmids. So bacteria has both of those. And on this plasmid, there is tumor growing gene. So bacteria takes out this gene and gives it to plant. And plant produces tumors for bacteria, little nice home. But along in the 70s, came a bunch of scientists and they said, wait, I can use that. So, they have, for example, fluorescence gene. And they took this plasmid and cut out the tumor part and put fluorescence gene. And they have this plasmid, they gave it back to Agrobacterium, and Agrobacterium gives it to plant. Plant starts glowing. Great. It's worth noting that different kinds of organisms have different methods of genetic transformation. For example, for plants, we scientists usually use Agrobacterium. It's very easy. So, the biggest question you can ask now is why? I know you can think it's like for making some nice pictures, cool photos. You could see those photos like outside during the break on the walls. But really, we use it for research. And all those reasons I gave it here, they amount to one word: visualization. Usually, when we scientists look under the microscope, we don't really see anything. Most of the cells, microscopic slides, are see-through. Until we give them some coloring or anything, we cannot see anything. So we use those fluorescent proteins to see structures, to see localizations of different proteins, of different processes in cells. So that's why we use fluorescence. So, returning to the main topic, how to make fluorescent fungus. And why do I care about it? Because it was my master's thesis. So yeah, we in our lab, we research Fusarium oxysporum forma specialis linii. It's a long name, you don't have to remember it. It's a fungi that attacks flax plants. For example, fungi like molds can attack humans, but this one just likes to attack plants. So we wanted to see how it enters plant, how infection goes in the plant, from where it starts, how it goes, how fungi grows inside. So we needed fluorescent fungi. So later we chose fluorescent protein, DS red in my case, but really nowadays there are fluorescent proteins in any color you want, yellow, blue, green, anything. So I chose the red one. Later we put this gene into agrobacterium and let agrobacterium do the work. But some of you may notice that I contradicted myself, because I said that agrobacterium transformation is used for plants and fungi is not a plant, it's totally different kingdom. So how do we do this? We trick agrobacterium. Here you can see this mysterious compound. It's called acetosyringon. This compound is produced by the plant when it's injured. What does it mean? So, when you have agrobacterium, and for example, our fungi, inside the flask, we add acetosyringon. And now, agrobacterium thinks, oh my god, there's injured plant nearby. I can make a new home for myself. I need to make transformation happen. So, we trick it into thinking that there is plant nearby, and it attacks and it transforms fungi. So now we get fluorescent fungi. So now you can see how a microscopic slide from flax that was attacked by Fusarium sporum forma specialis look without fluorescence. If you don't know like plant anatomy or anything, you cannot really see anything in here. I don't blame you, I didn't even see it myself. But after we use fluorescence, we get glowing results. We can see exactly how the fungus enters. And I hope you enjoyed my presentation and I interested you in fluorescence, maybe genetics, or a little bit of physics. Thank you.
Thank you so much, Anya. It's a very interesting presentation, I think. And also, I think the nature is truly beautiful. And nature has many fascinating functions, processes that we as biotechnologists studying, describing, and then we can use those processes for ourselves, and not only for the research, but also for helping people and other species like plants, anyone, I think, and all planet. That's our major goal. So, and that's why I think that biotechnologists are just magicians, because it's true magic, I think. And of course, I don't have any question because I'm familiar with those techniques, but I think you have. So please ask questions.
Please. I do.
Wow, our sound man.
Yep.
Oh, sorry.
My question is, in nature, what is the purpose of, for some plants or for some fungus or even for some animals to glow?
From what I remember, for example, there are these bacteria that, for example, use fluorescence to communicate with each other, for example. It's also like the way, from what I remember, sometimes it's just byproduct. For example, in plants, there is this process called autofluorescence. The plants have these things in their cell walls that just glow by themselves, and it's very awful when we try to get some nice photos, because plant glows, fungi glows, everything glows, we don't know which is which. So yeah.
Thank you.
Thank you. So my question is, are some plants became more vulnerable because genes were changed? So can we have some cons, negative effects during this transformation to then to become more fluorescence or something like this?
Or
plants not in danger. So my question,
yes, plants are not really in danger, but we have to remember that in Europe, using genetically modified plants outside laboratory is forbidden. So for now, we cannot really use those plants, but we're hoping someday we can, because usually it would help. For example, fluorescence is mostly used for research, but there are also plants that get some additional copies of the positive things, for example, more of vitamins or something else. And we're slowly developing methods until we're waiting for the law to be a little loosened so we can use those plants, so they can help people. And during developing of those methods, we develop them in the way so nothing like bad would stay in the plant. We only give the nice part. So yes, but in case, for example, of the natural transformation by agrobacterium, is the process that attacks the plant. So we scientists use different kind of agrobacterium. That the one that is, like, let's to say, non-pathogenic, let's to say, so it doesn't leave those tumors or nothing bad. So.
Fluorescent proteins of different colors have similar, for example, the red one and blue one have similar structure or totally different structure?
Mostly fluorescent proteins have different structure, but they usually sometimes have like the same. They have to, like, we call those motifs. So the same motif has to appear so it can get fluorescence. So usually proteins are different, but if you want them to have the similar function, usually those motifs will repeat in them. So sometimes they have those parts that are the same, but with proteins it can be really anything. We can get all those kinds of them. So yeah.
Thanks. So I wanted just to clarify for myself. So during your presentation, you showed us that in nature, also in science, if you wanna make plant glowing, you would transmit a gene from something which glows to this new plant, for instance. And then you showed us that during previous question, we could see that you also use proteins to make it happen. So actually, I have two questions. First of all, why proteins, right? Is it a source of genes, or I misconcept something?
Yeah, so in biology, there is this dogmatic view, let's to say, that it's like DNA, then it's RNA, and then from RNA we get proteins. So it usually, as dogmatics say, it only goes in one way. Of course, there are some exceptions sometimes, but usually from DNA, this is the recipe. We get then the copy that goes into the cell, and using this transcription of DNA, because we don't want to lose the DNA, we produce protein. So this is like the last part of the equation, this protein that is produced. And for example, we don't want to put just protein in the cell, because when we put something new into cell, the cell will try to metabolize it. It will try to make it like, let's to say, food for itself. So if we put just the protein inside the cell, it would destroy it. But if we, let's say, convinced by putting it in DNA, the cell will think, yes, this is mine, this is my gene, so I will produce it.
Okay, thanks. So in which case, if I would go to a sports shop and buy some protein, if I wanted to glow in the night, which protein should I use for this?
I think that's a hard question, because I think you would digest it in your in your intestines. So I don't think it would glow. You wouldn't glow. I'm sorry.
That's such a pity. Thank you.
But we can transform you if you want.
Yeah.
Yeah, some injection. You want?
Can you do something that I would not be digesting all night long after?
I don't know. We can think about it.
This will be your home task.
Thank you. Okay, so maybe questions online? Someone want to ask something? No? Anyone? No? Okay. So, we will continue with our last presentation of this